Okay, so welcome to this next video on the Human Genome Project. In this video, what we're going to discuss is Sanger, which is also known as capillary sequencing. So we're actually now going to discuss how we sequence these fragments which we've put in this genome library. So just to recap, what we are trying to do, remember, is we are trying to sequence the organic base sequence of chromosome 1 to 23. We cannot just perform Sanger sequencing on an entire chromosome. They are too big. So instead what we have to do is we have to fragment it down into small pieces. The problem then occurs, is how do we put the pieces back together? Well, to overcome that, we came up with this idea of restriction mapping, which was this idea of get multiple copies of the chromosome 1, or any of the 23 chromosomes, and then perform a partial restriction digest, where you only put enough restriction endonuclease to cut a few of the sites, and not all of them. So, what will happen is on your different copies of chromosome 1, you will get the chromosome being cut at different restriction sites, basically. So you'll get different fragments being produced. And then, potentially what you will have is overlapping fragments. And from that, you can then work out how to patch them back together and get the full sequence for the chromosome. So, what we have done is we've taken our multiple copies of chromosome 1 to 23 and we have now done a partial restriction digest. We've got these fragments and we've put each fragment into a vector, basically, into a plasmid. And then what we've done is we put each one of these plasmid into its own bacterium, which has created us our genome library. The bacteria then copy the plasmid, so each bacterium has multiple copies of this plasmid which contains one of these fragments from our partial restriction digest. Okay, so now all we need to do is sequence the fragments, each fragment in each of these bacteria in our genome library, and this is why it took 13 years and £3 billion, because you're going to have a lot of fragments and a lot of bacteria, and it's a very long and tedious process. So, let me explain how what we used to do, the way we used to sequence these fragments, and this was a mechanism known as Sanger or capillary sequencing, and I think I'll get a new piece of paper to start this with. Right, okay. So our starting point now is a bacterium which has multiple copies of this plasmid in, which contains one of our fragments from the partial restriction digest. Okay, so these are all utterly identical plasmids. Now, we are going to perform the process known as Sanger or capillary sequencing. So Sanger sequencing is the topic for this video now. Right. And the principle is this. We are going to harvest these plasmids from the bacterium, firstly. So we're going to take all of these plasmids out of the bacterium and separate them off from all of the other goo inside the bacterium. So we've got all of these plasmids outside of the bacterium now. Okay, and they all contain this fragment of the genome, basically, one of these fragments that came from the restriction digest. Now, we have set up this plasmid. When we put the fragment inside the plasmid, we didn't just pick any old plasmid. We picked a plasmid with a certain region here to which we can design a nice primer. So basically, if I draw a bigger picture of this plasmid. So I'm going to draw it with double-stranded DNA now. So I'm going to draw both DNA strands. So let's say this is our plasmid. Okay, and this is the second DNA strand to this plasmid. Okay, right. So some region of this is our fragment, our DNA fragment. So let's say here is our DNA fragment in here. Now basically, in front of the DNA fragment is a certain region which we will design a primer to, basically. So, let's say we take this region here. Whoops, let me just deal with this problem of me shaking the whole thing around. Okay, so we're going to take a certain region here and design a primer to it, basically. So now, let me draw this out linearly so that we can have a look, and let me give you some examples of organic bases that we could put in there. So let's just sort of bring this bit out and have a look at it in linear view. Okay, so I'm just bringing part of the plasmid out, and we're just looking at it, but I'm not actually taking it out as part of the experiment. It's still in the plasmid as far as the experiment's concerned. We're just bringing it out to have a look at it. Okay, so here's this portion to which we are going to design a primer, and here is our fragment that we've put in. Okay, so this primer region will be a sequence of maybe 8 to 10 organic bases to which we are going to design a piece of DNA which is complementary to it. Okay, so let's say it has some organic bases here. Let's say it has the sequence A, C, G, and you could go on, etc. So it might have 8 to 10 organic bases. You wouldn't just use 4, that would be not specific enough. You'd need around 8 to 10 organic bases. Right, now what we want to do is design a primer to that, which is basically going to provide us with a starting point from which DNA polymerase can work. So I think actually rather than go into the methodology at the moment, I'm just going to outline the basic principle of what Sanger sequencing is going to do, and then I'll go into the details. So the idea is this. We are going to take these different plasmids here, these multiple plasmids which all have this same fragment in, and basically what we're going to do is we're going to set up a DNA polymerase basically copying this fragment, but in the medium in which this DNA polymerase is operating, we are going to put in 2 types of nucleotide. We're going to put in working nucleotides for all 4 organic bases, so adenine, thymine, cytosine, and guanine. We're going to put all of those in, but then we're also going to put terminator nucleotides for every single one of the 4 organic bases. Okay, then what we're going to do is we're going to let DNA polymerase work, and it's got multiple copies to work on. So it's going to start copying these fragments basically, but every so often, instead of putting in a normal nucleotide, it will put in a terminator nucleotide, and then basically it will have to stop copying the DNA. So the copying of the DNA sequence will stop prematurely basically. Okay, and we can then look at the different lengths of these different fragments, and from that we can work out what organic base was at each position, because what we will do is we will fluorescently label each of the terminator nucleotides with a different color. So by looking at the color that each fragment is releasing, and also looking at the length of it, we can work out at each position what organic base was there. That's the basic principle. I'm going to describe it in detail now. So, in essence, we don't need 2 strands of DNA. We have 2 strands of DNA, but in fact we only need to sequence one of them. As soon as we know the sequence of one, we instantly know the sequence of the other. So let's say we're going to sequence this one here. So the other one is basically surplus to requirements now. We don't give a damn about that one basically anymore. Okay, so one of the first things we're going to do is heat up all our plasmids. So we're going to heat up all our plasmids to around 90 degrees, so to around 90 degrees Celsius. And the reason we're going to do this is to break all the hydrogen bonds between the complementary organic bases on both strands. So what this is going to do is it's going to cause both strands to fall apart. So what's going to happen is that this plasmid, which was double-stranded, is going to fall apart into 2 single-stranded pieces of DNA, like so. Whoops, what have I done? Okay, there we go. Into 2 single-stranded pieces of DNA. So, let me highlight everything back in again. So here's this pink region from here. Let's say this is our green region here. Okay, and then this is the other strand which we don't care about. So we've heated up to 90 degrees and we've now broken the hydrogen bonds. What we are now going to do is add in to the medium primers which are complementary to this pink region of the DNA basically. So what we will have done, if I pull this single strand out now, so let's say we've got rid of this one here now, and we've got this pink region here. Okay, and this time I'm making it bigger so that I can actually put 8 organic bases on there. So let's say its sequence of organic bases was A, C, G, and I'm making this totally up. A, T, C, T, like so. Okay, so this pink region just happens to have that sequence of organic bases, and it's upstream basically of this green region which we are trying to copy. Okay, right. So what's going to happen is we're going to create a primer to this basically. We're going to create a big portion of DNA which has the exact, which has the complementary organic bases to these. So it will have thymine here, which is complementary to adenine, thymine here, guanine here, complementary to cytosine, cytosine here, complementary to guanine, thymine here, complementary to adenine, adenine here, complementary to thymine, guanine here, complementary to cytosine, and adenine here. Now this strand, this 8 organic base long strand, this is our primer basically that I'm colouring now in turquoise, and it does not go beyond that. It's just these 8 organic bases together basically, so it's a short piece of DNA. So this is our primer. Okay, so what will happen next? Okay, so we've heated our plasmids all up to 90 degrees, so the 2 complementary double strands of DNA have fallen apart basically. The hydrogen bonds have cleaved. Now what we do is we add in our primers. So our primers bind to this pink region on our on this strand of DNA here, but not onto this one, because this one, remember, would have had this same sequence here as the primer has, so it can't bind to it basically. Okay, and we'll continue this discussion in the next video.